now. So, welcome. We have people from De Novo as well as Gemini Bio, and they're going to present a little bit of information for us today in our cell culture class. So, can I take it away and let you introduce yourself a little bit more for the students?
Yeah, absolutely. Thanks, Denise. My name is Sean Burke. I'm a director of research product development here at De Novo Software. We make a software program for analyzing flow cytometry data sets. So I understand that there may be a little bit of flow cytometry going on tomorrow from some of the cells that are being grown up. And essentially what I've been tasked with today is to give you a little bit of a primer about flow cytometry in general. It's really a kind of a broad field, and I'm going to be going through a little bit of that, some data analysis techniques, and of course a little bit about how to use our software for analyzing some of the data. So with that, you want me to take it away here, Denise?
Sure. Or unless
All right.
George wants to say something. George, you're hiding behind a black screen. I don't know if you want to say anything. You're coming in tomorrow, so you might want to introduce yourself.
Hi, everyone. My name is George Hung. I'm the West Coast Field Application Specialist, and I think we'll be learning more about different types of roles in the space toward the end of the class. I'll be coming in tomorrow to assist with an experiment that we have. We have a couple of other members on our team as well that are just that are here. Richard Simmons and Andrew Jareski. Excuse me about the last names. But I'll turn it over to Sean so that he can kind of get started. But hope you all enjoy this, and we'll definitely look for some feedback from you all toward the end of the session.
Thank you.
Excellent. All right, so I think I'll just dive right in here. Right, again, kind of as I mentioned, what I really want to cover today is just what is flow cytometry at a very high level. I know a lot of folks here might not even know what a flow cytometer is at this point, right? So we're going to kind of get you familiar, and that way when you see these things and you go into labs, you hear about people using them, you're reading publications, you have a little bit of kind of base knowledge to start, which I think is very helpful. We'll go over some of the data analysis basics. So when we take data off that instrument, what do we do with it? And then we're going to come in and actually look at some real world data in the software that our company produces. So I think one place to start is kind of why flow cytometry. I got into this field maybe 10 or 15 years ago when I was a researcher back in the lab. And it's really a cool technique. I mean, really what it is, it's a means of identifying and measuring all sorts of different things about cells or particles. And essentially what we do is we take cells, we put them in a test tube, we stick that test tube onto an instrument called the flow cytometer, and that instrument will allow us to rapidly take measurements of lots of different characteristics of that cell at the same time. And we do that, I say, on a cell, but in that test tube there can be 100 cells, 1,000 cells, 10 million cells. So it really gives us the power of looking at all of these kind of different markers, things chemicals and dyes that we add onto cells, at not just a single cell level, but at a kind of like big population level. So we get a real kind of broad picture about what's going on. So essentially, again, these cells are tagged with some sort of fluorescent molecule, and that cell is going to pass a laser, which is going to shine on that cell, and then light is going to be emitted from the cell, and we're going to have detectors that are going to collect this stuff. And in the end, there's a lot of different applications. We can look at fluorescent intensities of these markers that are attached to cells. We can look at immunophenotypes of cells. So we can look at, take a blood cell, and we can figure out, does that cell have CD3 on it? Does it have CD4 on it? Does it have CD8 on it? Does it have all of these things? If we treat them with a drug, do those markers go up? Do they go down? Right? So there's lots and lots of applications for these. But I think the most important thing to kind of get out of this, and really any technique, is flow cytometry is just a tool in a scientist's toolbox, right? But the thing about flow cytometry is it's widely used and it's encountered in a wide range of fields. So, whether you're doing, basic biology, you're doing microbiology, you're working with bacteria, you're working with yeast, you're even mental health. I've seen, people use flow cytometry to make measurements of blood in mental health studies and things like that, right? You're really going to see it. It permeates everything. So just knowing a little bit about it, as you enter into these fields in your careers is going to be a really powerful thing. So first, I just want to give you a little bit of an overview of kind of what flow cytometry does, right? And what these instruments look like. I'm going to kind of get down in a more granular level as we go along here. But essentially, we're going to take cells, right? In this case, we've got some different cell types. There's a killer T cell that's got CD8 on it. We've got a helper T cell here that's got CD4 on it. And what we need to do in flow cytometry is tag these cells in some sort of a way, right? So, I don't know how much you've learned about antibodies yet, but you can get antibodies. There's manufacturers and companies that make these things that are specific to target cells that have CD4 on them or target cells that have CD8. So if you add these little antibodies in with your cells in solution, they stick to the cells. And what we can do with these antibodies is we can also put fluorescent markers on them, like literally these markers that turn your cells green or turn your cells red or turn them blue or really all sorts of different wavelengths of light. And what I'm kind of showing here on the left is just antibodies, but it doesn't just have to be antibodies, right? It can be chemicals that we add into your cells that get incorporated into the cell and they fluoresce. You've probably heard about things like green fluorescent protein and jellyfish and bioluminescence, right? That sort of kind of bioluminescence can be incorporated into cells and it can be expressed from their DNA in certain ways. And we can measure that with a flow cytometer, right? So there's lots of different ways that you can get these cells to fluoresce. But what will happen is that cell gets put into a test tube, or 100,000 or 1,000,000 cells get put into a test tube, and you stick that onto your flow cytometer, and the fluid from that tube gets sucked up into the machine, right? It goes from this little sample here, gets sucked up into what we call the sheath or the flow cell, and these cells will pass a laser one by one, and as the cells pass the laser is going to shine light on that cell. The fluorescent tags are going to fluoresce different colors, and that's going to go into a whole series of detectors. That data is going to get stored in a computer, and in the end, every single cell is going to turn into a little dot on charts, right? And these little charts that we're looking at on the right-hand side, there's all sorts of little dots in here. Each one represents an individual cell and a measurement that we've taken from that cell, right? So there's lots of different types of flow cytometers. When you go out into the lab, and you're going to see one of these probably tomorrow, but they come in many different shapes and sizes and configurations. You have some really handy stuff here, like this little handheld cytometer. This one down here, you can see the person's hand in there, so it's really kind of a tiny thing, right? And they really range up to cytometers that are the size of a desk, the size of a large desk, right? The size of that hood that you were working in today or yesterday, and really depending on what you need is what type of cytometer you're going to pick, right? So imagine that you're on a boat and you need to analyze algae somewhere in the middle of the ocean. Well, you're not going to bring one of these big benchtop things out there, right? It's going to be a major pain in the butt. But getting something like this little kind of handheld one is going to be really helpful, right? It's also going to depend on how many simultaneous markers you need to collect, right? Do we need to look at CD4 in green and CD8 in red, or do we need to do those and then 5 other ones or 10 other markers, right? So the more markers that you add in there, the kind of more detectors, and the bigger that these kind of boxes are going to be able to come in. So long and short, you're going to see a lot of different types of cytometers out there, especially as you go into the field and you become biologists and scientists. But they all kind of do the same thing in the background to one degree or another. So kind of jumping into a flow cytometer, just a little bit closer look of what's going on. I mentioned those cells from your sample are going to go into the cytometer, and they kind of come through like this, right? They go through, you might get like a kind of big concentration of cells in that fluid kind of going into your cytometer. And the cytometer does some really cool things to turn that kind of big jumble of cells into a single stream of cells that can go past a laser. And that laser is going to hit each individual cell as that cell goes past the laser. Now, when the laser hits that cell, it's going to send light two different ways. One is it's going to send light directly through that cell, and we have a detector over here called FSC, and that's forward scatter. It means light that's simply passed directly through that cell, right? And then light is also going to bounce off of that cell. And in particular, light that fluoresces in different wavelengths is going to bounce off to the side. And we call those kind of side scatter detectors, or SSC, and we measure different wavelengths of light with a bunch of other kind of array of detectors. So, some of the kind of smaller handheld cytometers might only have one or two of these little detectors here, but some of the bigger, ones that are designed for measuring 10, 20, 30 different markers at a time are going to have banks and banks and banks of these kind of in different configurations. So essentially what happens is the light hits that cell, information about how much light at different wavelengths goes out to all these different detectors. There's a whole bunch of kind of electronic processing that goes on, and then all that information is sent to a computer and stored. And I think the really cool thing about flow cytometer, or cytometry in general, which you can kind of see here, is that there's so many sciences that become involved in this, right? We have chemistry, we have for putting together chemicals and antibody combinations, we have biology from the cells themselves, we have fluidic systems, we have physics and optics, we have electronics, right? So these instruments are really rather complex, right? They can be simplified into little handheld tools, which are really, really great. But the more kind of complex and the more markers you get, these things can get very complex. And there's entire, week, month-long courses devoted to, learning how to build a flow cytometer and how all these things come together. But the good news for all of us as scientists and future scientists is that there's generally facilities within universities and research labs that have trained people on staff to work these things. And, if you have an experiment in mind that you want to do, they can tell you how to set that up, how to bring the cells to them. They run everything on the cytometer, and then they give you the data on the back end, and you can go back to your lab and analyze that. So those are kind of called core facilities or shared resource facilities. And they're really a fantastic tool when you start, kind of getting out there into labs. Now, the other kind of thing that a flow cytometer can do is, when those cells kind of come through here, past this laser, I only kind of showed, right, they kind of end up down here when they go past the laser. What actually happens? Well, when we're just analyzing those cells, where we're just trying to make kind of static measurements of what's going on, they just go into a waste bin, right? They usually end up into a little bin that has some bleach in it to kill the cells, so nothing grows, and all the kind of bad bacteria and viruses that you might have treated them with kind of all die and get killed. But there are certain cases where we use something called a cell sorter. It's a flow cytometer that can actually take those cells, and instead of just throwing them away at the end, it can sort them into different test tubes, right? So imagine that you've treated your cells so that, some cells turn green when a certain gene is expressed, and some cells turn blue when another gene is expressed. Well, these sorters are pretty darn cool, because as you're running these cells through this laser and through this system, the acquisition software, the software you use to make those measurements, can come back around, and by the time it gets to the bottom of this little kind of sheath fluid area, it can charge the droplets that cell is traveling in, and with a little charge applied to them, if it has a negative charge to that droplet, well, they can put a little kind of panel with a positive charge, and those cells are going to be kind of pushed in one way as they fall out of the cytometer. And if you assign cells a different kind of positive charge, and you have a negative plate, right, they can be pulled out to another side. So instead of just throwing those cells away, which, is not a bad thing in a lot of cases, well, you can actually take a whole bunch of cells that are combined into a fluid, and you can sort them out, right? And you can say, take the ones that express this really important gene of interest, put them into this tube and throw everything else away, and then grow up those cells and do something interesting experimentally with them. So a cell sorter is just kind of another type of cytometer that you might see out there, and it's another really important tool in the scientist toolkit. Now, when I kind of showed, this slide earlier of all these kind of little detectors, there's these little, kind of blocks that are in here. And what those blocks actually are filters and mirrors. And essentially what happens in a cytometer is when that cell is excited, right? When the laser shines on that and, a red signal from that cell is excited and it's kind of passed on to the detectors, or a green signal in that cell is excited and it's passed on to the detectors, we need to figure out a way that we can assign that color to a specific detector. We can't just send it to one detector and then it figures out what all the different colors are. And what we do in a cytometer is we use these little filters and mirrors that allow us to allow the green light to kind of pass through this filter, and when it gets to the specific type of mirror, bounce that green light down here to a green detector. But if there's red light on that cell as well, let that red light actually go through that filter and then bounce down into another detector here. And this is actually pretty applicable across a lot of different fields of science, not just flow cytometry. It's things that you use in microscopy. There's things like this that are used in all sorts of detection platforms. And what these kind of filters and mirrors do is they work in a few ways. One is there's something called a bandpass. And really what this allows this filter to do is that if light of a certain wavelength, here it's called the 620 to 640 bandpass filter. So if light, which is kind of in that red range of 620 to 640, well, that will flow through that mirror or that filter and go on to the next thing. You have things called long pass filters, where if a light is above a certain wavelength, it will go through the filter, and if it's below that, it will get blocked by it. Things called short pass filters, where if the light is below a certain wavelength, it will go through it or it will get blocked. And also something called dichroic mirrors, right? And what these do is they allow light of a certain wavelength to go through it, and light that doesn't meet that wavelength to bounce back off of it. And again, that's kind of what we get here. We use a filter that allows some light to go through. We use a set of dichroic mirrors that allows light of certain wavelengths to go through. But when it reaches a dichroic mirror that's going to reflect all the green light, well, that's going to go into the green filter. And when it reaches another mirror that's going to reflect just all the red light, that's going to go into another detector. Right? So, with kind of more simple systems, you might just have one or two of these kind of little filters or mirrors in here. But when you're working with flow cytometers that collect 18, 19, 20, 40 parameters or 40 different channels of markers, there's a really complex system of optics and physics that are going on in the background. But you might be wondering, all right, well, why do you need filters and mirrors? Why don't we just collect everything, right? And there's, some very specific reasons with flow cytometry why we can't just collect everything. When it comes down to it, we look at what we call excitation and emission spectras all the time. And if you haven't had the physics class on this, you're going to have the physics class on it. You're going to learn about this in a lot of detail. But really the idea is when a light of a certain color, say green, emits, it doesn't emit in kind of like one wavelength of light. It will actually emit here. You can see this kind of green curve. It's going to emit from somewhere around 450 nanometers to up to about 650, right? So it's actually a very broad range of light that's being produced. And things that fluoresce in the red channel, and I say red, this is kind of yellow here, this yellow one, it's going to fluoresce from kind of 550 to 650 and beyond. So if we collect all the light, we're going to be collecting things that are red and things that are green. So we have to use these kind of special filters to come in and just grab the portion of the light from green that we know is green and the portion of the light from, say, yellow or red that we know is yellow or red. Right. I'm going to come back and kind of talk about that in just a little bit. But how is this data collected, right? I say it goes into a detector. Well, what do we actually detect? Well, when that cell passes a laser, that cell gets excited, right? The light inside of it gets excited. And the machine or the computer makes a measurement of the intensity of that light, which you can kind of see here on the y-axis, across time, which is how long it takes for that cell to go past that laser. And that results in something that we call a pulse area, right? And it's just, that's the measurement. The pulse area is just the measurement, how much light came off of this cell when it went past the laser. But you can see it's kind of like this little curve thing, right? And if you have a curve like this, you can make different measurements. You can measure how high that curve is or how high that peak is. You can measure how wide it is. You can measure the area under that curve. And in fact, flow cytometers generally make all three of those measurements. And we use them in different ways when we analyze the data kind of down the line. And essentially for every signal that you get, if you have a green signal and a red signal and a blue signal, we're going to get different pulse areas for all of those for every single cell that you're analyzing. But what happens in reality? I show some of these kind of diagrams and figures. I'm sure you see these things in your books, and as you proceed to become scientists, companies are going to give you diagrams and figures to look at. Well, that isn't always what happens in the real world, right? But you see a diagram and a figure, all the cells pass single file, pass a laser. That looks awesome, right? Well, what happens is cells are sticky. They clump together. You're probably going to find this when you start working in cell culture, that you need to kind of get cells that come off the bottom of the things that you're growing them on, or when you grow them in suspension, they all like to stick together. Well, in a cytometer, that still happens, right? And we need to find ways to kind of pull these cells apart so that way they go past our laser one at a time. And I think one of the most important things when I learned about cytometry in the beginning is really clumping and those cells sticking together, which you don't want when you run flow cytometry. It can really be minimized by experimental protocol, right? And what that means is kind of this, this old saying of garbage in, garbage out, right? If you don't, take the time to follow the directions and follow the protocol in your experiment correctly, you're going to give garbage kind of information into your cytometer, and the data that comes out is not going to look so great as well. So if your protocol says, chill your cells to 32 degrees and spin them for 37 seconds and, tilt your head to the side while you're shaking the tube, somebody probably did that and it worked out for some reason, right? So make sure you read those protocols, you follow them exactly as they're written, and that's going to really help save you a lot of time and a lot of frustration, actually. If it takes a day or a week to do an experiment and you get to the cytometer and all the cells are clumped together and you got to go do it again, that's a real big bummer. But the reason that this is kind of in particular a problem is you kind of ignore these G1, G2 things up here, but if you take a cell and it has a certain signal and say that value equals 1 when you run it past the laser, well, if you have two cells that are stuck together that have a value of 1, when it runs past that laser in that detector, the detector is going to see a value of 2 because it's going to take an additive measurement of the two cells that are stuck together instead of a measurement of just one cell at a time. And it doesn't just happen in 1 or 2 cells. It can be 3, 4, 5, 10, 100 cells can stick together. If they get too sticky and too clumpy, they will just kind of clog up your flow cytometer and it won't run. But they can make your data intrinsically very different than it should be if you're just running single cells. So in the end, take the time to read the protocols, do the experiment correctly. If you're working with one of these core or shared resource facilities, they're going to give you some advice on how to kind of do this. If you're working with that data afterwards, if there is some minimal clumping, there are ways that we can use software to kind of help detect what is a clump of cells versus what is a single cell, but that only goes so far, right? Garbage in, garbage out. So again, take the time to make sure you're getting your experiments and running them in the protocols that are designed, and you're not going to run into any problems. So, what does this kind of look like when you put it all together in the end? What types of information can you gather, right? I got a kind of it's not a super long list here, but this list could be, pages and pages long of the types of information that you can gather. You can count cells, you can look at cell characteristics and functions, you can detect biomarkers, you can look at proteins going up and down, diagnosis of health disorders. A lot of cancers are diagnosed using flow cytometry. You can look at how cells proliferate, how cells progress through cell cycle, how they live and die, right? So that list, again, just goes kind of on and on and on, and that's why flow cytometry really permeates a lot of what you're going to be doing in the sciences. But I did just want to give you kind of 2 real-world examples here before we talk about data analysis a little bit more, and I'll give you your first kind of glance of what this data looks like when it comes off of a cytometer. So this is some data from the instrument that you'll be using tomorrow. And essentially what we have here, you can see there's a graph, and we're counting the number of cells on the y-axis, and an x-axis here we're looking at FL fluorescence, and that's the intensity measurement of the light that went to that detector on the cytometer, right? And I've got a whole bunch of kind of different files here, right, in what I call data list in FCS Express. And as I click on them, you can kind of see things over here changing, right? So each one of these is its own test tube. And if I click through this, it's giving me some sort of measurement. Now, these cells were stained with propydian iodide, right? This isn't one of those antibodies. This is a chemical. And if we put propydian iodide in a tube of cells, propydian iodide only sticks to cells that are essentially dead or dying because it can't permeate the cell membrane, right? So things that are dead and dying, the cell membrane is degrading, it's not there, it's going away, and propydian iodide can stain that. So things that have a high fluorescence over here are generally considered dead, and things that are, alive and have a very low fluorescence are considered live over here. So again, what we can do with a very simple experiment with flow cytometry is treat cells with something, maybe it kills them or makes them better, right? We want to see how they die or live one way or the other. And then we can use tools like our data analysis software, FCS Express here, to kind of navigate through these data files. We can say, all right, well, here's 11% have died, 17% have died. Oh, well, nothing died over here, right? Only 0.13% of the cells have died. So, what happened with that sample, right? What did we treat it with to get cells to live, right? If you're doing an experiment, you want to cure cancer, well, you want to kill cancer cells, right? You want cancer to die, right? And you want more dead cells over here. But if you're doing some sort of other experiment and you're looking at maybe immune cells in your blood, well, maybe you want those immune cells to live, right? So you want to make sure that you're running your experiment so you can determine how many of those cells live, right? So this information, it's not kind of giving you one piece of empirical information here. It's just telling you what's live or dead, and depending on the experiment that you're doing, it's up to you to kind of interpret what's going on there. Also, kind of importantly, you can see I've got a list of all of the cells that were run in the cytometer, right? So this kind of histogram, which I'm going to talk about in a little bit, is just a summary of all the measurements of all the cells, right? So, I don't just have a few cells. I ran, somewhere like 5,000 or 6,000 cells through the cytometer, 4,055 cells, to gather these measurements, right? So instead of just looking at one or two cells like you might do in a microscope, a flow cytometer allows you to look at a really big population of cells and gather some information. Now, in this case, we're just looking at one marker, right? It's just one, marker for propydian iodide. But as I mentioned, the cytometers can get really big and they can get really complicated, and that's not a bad thing because they can give us a lot of really useful information. Now, in this other experiment, this is what we call an immunophenotyping experiment, where we take, say, blood out of somebody's body, and we isolate all of the white blood cells from that tube of blood. Again, if you, heaven forbid, you have to go get a cancer test or something like this, they might do that. They take blood from your body, they isolate all the white blood cells, and then they put markers on those cells for things that identify what's a T cell, like CD3, things that identify what's a helper T cell, like CD4, things that identify what's a natural killer T cell, things that identify what's a B cell, things that identify what's a dendritic cell, right? And in this cytometer, we can actually measure up to 33 of these different markers on each individual cell. And that allows us to kind of go through and generate these really complex reports where we can say, all right, for all of our T cells, here's how many natural killers, how many are cytotoxic, how many are helper T cells. For dendritic cells, how many are, macrophages, how many are, other dendritic cells. So we can break down all these kind of individual markers, and we do so by kind of using all of these graphs that are available to us in the data analysis to evaluate what these cells are expressing, how they're expressing it, and how much they're expressing it. And that's what our tool, FCS Express, allows people to do, is take this really kind of complicated information that comes off of a flow cytometer and distill it into some sort of way that even a clinician or a physician can come in and look at this report that we put together, and they can say, all right, well, based on the amount of CD3 and NK T cells and helper T cells that you have, you're healthy. Or if those values change, maybe you have cancer, maybe you have some other sickness in your immune system. And that's how, again, flow cytometry is used kind of in a clinical sense to kind of make evaluations about what's going on in a patient's immune system. But just as well, I mean, you might be treating mice in the lab with a drug treatment, and you want to see what's going on in their immune system, right? It's not kind of a clinical test; it's kind of a research test, and these tools are really helpful and functional for that. So that's a little bit about, how we get to this data. But you might be looking at these plots and thinking like, oh, well, what the heck is that, right? That looks like a pretty cool looking, red and blue plot with a bunch of dots in there. What does that actually mean, right? So that's where I'm going to kind of skip over here, and we're going to talk a little bit about when that data comes off of the instrument. Once it gets to that computer workstation, well, how do we get to these kind of plots? What do they mean? How do we interpret them? And even, maybe how do we do a little bit of this on our own, right? So one thing to kind of understand is that data is stored in what's called a FCS file. That's what the instrument creates, right? And an FCS file is just like a file on your computer. Like you have a PDF file or a PowerPoint or a TIFF image or a PNG. It's just a type of file, and that's what's stored by a flow cytometer. There's a whole group of smart people, or people who think they're very smart people, who create standards around these files and what they do and what they store. But I think for the purposes of today, there's really two important pieces of information in these data files. Something that's called text or keywords, and the data, the values, the measurements that come from individual cells. And they both have important places in the science. So the keywords are essentially ways that the instrument that you're acquiring data on stores information in a text type of form, right? So what day you acquired that experiment, who acquired the experiment, what did you treat these cells with that day, right? And all that information gets stored with the data file. And I think this is really important to everything you do in science, is to make sure you annotate your data, no matter whether you're doing flow cytometry or you're just doing something, in your lab notebook, writing down about, what you treated the cells with that day. Annotate your data as best as you possibly can. And especially with an FCS or a flow cytometer, because a lot of the times you're not collecting 1 or 2 or 3 data files, you're collecting many hundreds of tubes. And when you come back to that data 3 months from now, because you've been collecting hundreds of tubes for a few months, well, it's going to be really helpful if you can come back into the data file and you can see that, all right, Joe collected the data file, Jane analyzed it on this day, we treated it with, antibiotic resistant drug 1, 2, 3, and we did this, that, and the other thing. So these keywords are just a way to kind of annotate your data. The instruments do store useful information in there for helping software analyze it. But the thing to say about this is annotate your data. Don't forget to do that. It's one of the most important things that we can do as scientists and communicators, is to make sure all of that is done correctly. Now, when it comes to the data portion, what are we actually looking at? We're looking at measurements made on a per cell basis for whatever detectors we had in that system, right? And as I kind of mentioned, we have charts that kind of look like this, which we call 2D plots, and every kind of dot on that chart is a cell, right? It's an individual cell, and we've made a measurement about that. In this case, we made a measurement about how much blue it has and how much of another type of blue it has. But when we work with biological samples, we can do some interesting things where we can make some very dim measurements and we can make some very bright measurements and put that on the same graph, right? So you might have something that has a measurement of 1 and then something that has a measurement of 1,000,000. So when we do that, that's kind of hard to represent in a kind of 2-dimensional space, right? A kind of really wide range of data. So what we do with flow cytometry data is we have different ways to scale these plots. And when I say scale, you can see here on the y-axis, we're using a logarithmic scaling. We're going from 10 to the 3 to 10 to the 4th to 10 to the 5th. Instead of doing something, just saying, this is 1000, 1001, 1002, 1003, which would be a linear scale. But luckily for all of us, most software that analyzes this data is going to default to some sort of optimized way of looking at the data. But just so you get an idea of kind of what this looks like, I'm going to go over to FCS Express here, and we'll take a look at some real data, right? This is some cells. They were stained with CD8 on the y-axis here. They were stained with CD4 on the x-axis. So these are kind of intensity measurements of the amount of CD4. So things that are over here on the bottom right, they're very bright for CD4, but they don't have any CD8, right? And things that are over here in the upper left, they're very bright for CD8, but they don't have any CD4. Things down here, they're CD4 and CD8 negative, right? But the kind of optimized default display for this is what this chart looks like in the upper left here. Now, it's using this logarithmic scaling for most of the data. Because if we took that really wide range of data and we tried to put that on a linear scale, all that stuff is kind of squished down here in the bottom left of the chart. We can't really see what's going on. It's actually the same data. But because it's on this linear scale, everything is all smushed down and it's very difficult to see. But we could also put that on what's called a pure log scale, where we're just looking at 10 to the 0, 10 to the 1, 10 to the 2, 10 to the 3, and that data kind of gets stretched out, and it's easier for us to kind of digest and take in as scientists for this data. There's also all these kind of what we call hybrid scalings, where we can do special things, like things that are on the low end of the data, we put that on a linear scale because it kind of spreads out nicely there, and then as it gets more intense, we put it on a logarithmic scale. So those are called hybrid scales, and we don't have to worry about too much about what they are. But I think the most important thing here is that there's this little percentage up here. It says 15% of the cells fall within this little box I created on this chart, right? And you can see down here on this plot, well, there's 15% of the cells that fall within that little box too. Because this box and that box are the same box, but they're just distributed differently in the spaces of scaling that they're shown on this plot, right? So what I'm trying to tell you here is, it doesn't matter if you look at this on a linear scale or a log scale or one of these special scales, the data is always the same. What you're most interested in is getting this data into some sort of way that you can look at it visually as a scientist and say, well, that's my CD8 only population, that's my CD4 only population, and then I can look at how those populations change with treatments and different cell types and things like that. So again, software is going to optimize most of this for you, which is really, really nice. But don't get fooled, right? When you look at data, if it looks all cruddy like this and it's down in the bottom left-hand corner, the data might be really, really great. It's just not scaled in the correct way. And that can go for a lot of things that you're looking at in science as well. So another way of looking at data, and when I showed you that live dead staining, is something called a histogram. And how do we arrive at one of these histograms, right? So when we make a measurement on a flow cytometer, we measure cell number 1 that goes past that laser. And we made a measurement, in this case it's 0, right? We made, there's nothing on that cell. Then cell number 2 came along that laser. And we make a measurement, and the measurement is 256. So we can assign it to a value of 256, right? And we make a measurement for cell number 3, and a measurement for cell number 4, and a measurement for cell number 5, and another measurement for cell number 6. And it kind of looks like we're building up like little bar graphs here, right? And that's how we generate these kind of histogram displays that we look at with flow cytometry data. But we don't just do it with 6 cells, we do it with 6,000. Right? Or 6 million. So really what these histograms are a bar chart with, a bunch of bars, right? And when we look at data that ranges on a particular scale of, say, 0 to 1024, that means there's, 1024 bars that we can add data to. And when we look at flow data, we rarely draw every single bar, right? If we drew every single bar for 10,000 events in this little histogram here, the whole thing would look black and, like really kind of jumpy all over the place. So what we do is we connect the tops of the bars to the adjacent bar. And it arrives at these plots that kind of look a little bit smoother like this, right? So we can kind of see distributions between the data. And what this allows us to do is we can visualize the separation of negative populations and positive populations, things that don't have fluorescence or do have fluorescence. And we can assign, them to different phenotypes that we define. Are they live? Are they dead? Are they high? Are they low? And that's how we can kind of do some assessments, right? But when we look at those 2D plots, essentially all they are is histograms in 2 different dimensions, right? Instead of looking at just a histogram in 1 dimension with 1 axis, we can look at it in 2 different axes. And there's a lot of different ways to kind of visualize these 2D plots, right? So a lot of what I showed so far are just these little dot plots, right? Every dot is a cell, and that cell is a measurement, for whatever markers that we're looking at. But we can also give these kind of a topographical display by putting contours around them, like a contour, topographical map, where things that are kind of very contoured together, that's where the bulk of our data or the bulk of our populations are. Or we can give that some sort of density display, right? And in these cases, we're not plotting every individual cell. We're kind of plotting what's going on in that region for those cells of interest. So there's, again, a lot of different ways you can kind of visualize this stuff, but when we're looking at this data, if we're looking at a contour plot or a density plot or a dot plot, really underneath we're just representing the individual cells and where they fall as populations or groups of data. Now, when we create these charts, either histograms or 2D plots, we usually have to define what type of cell is in those kind of populations, and we do that by using something called a gate. Essentially, a gate is a way to define a population of interest that you're interested in looking at. So we can create, elliptical gates, kind of circles. We can create squares. We can create polygons. And it's kind of like just drawing objects in PowerPoint or in Photoshop or whatever. But what we can do is, let me kind of skip to the next one here, is that we can label those gates because we can know that, certain cell types of interest fall within these kind of gates, right? So if I'm looking at some of these side scatter versus forward scatter detectors, we know through the literature that, cells that kind of fall into these kind of lower areas are actually lymphocytes. Things that kind of get bigger are monocytes, and things that are bigger than that are granulocytes. So what we can do is we can create a little gate around the lymphocytes, and we can look at another chart of CD3 versus CD4 of only the cells that fall in there, and then we can see the expression of CD3 and CD4 for the lymphocytes. And then we can take the monocytes and we can put them into another graph of CD3 and CD4 and see that they only express CD4 but not CD3. Right? We can kind of do that over and over and over again through the populations of interest and kind of identify what's going on. Now, there's also something called backgating, right? And essentially what backgating is assigning colors to these populations on these charts. Right? So what I can do is I can give these kind of populations here different colors from my gates, and then when I look at different markers, it helps me visualize, all right, without having looking at the gate that's applied to a plot, I can look at all these populations on top of each other, and I can say, all right, well, the ones in red are my lymphocytes, and they look like this. The ones that are purple are my granulocytes, and they kind of look like this. The ones over here in blue are monocytes, and they look like this. But we can even set a gate on this chart over here where we have the CD3, CD4 population, which we did over there. We made that a pink gate. And we can show that back on this other chart of other parameters. We can say, all right, well, when we look at CD3 and CD4 together, those are actually lymphocytes. So there's a lot of kind of cool ways that we can use gates to analyze data and to kind of figure out what different populations do. And, you can see here we have 3 gates over here, we have a 4th gate over here, and there's a lot of kind of different combinations of gates that can be used. And essentially what we do is we use something called Boolean Gating, where if we have something like this where we have gate 1 and gate 2 drawn, if we say look at the intersection of gate 1 and gate 2, that's called an AND Boolean operator. And we're just looking at the events that fall in between there. If we do not gate 1 and gate 2, it's everything except that little intersection. If we do gate 1 or gate 2, it's things that fall within both of those gates. But we make this very simple in Flow software, because we use something called a gating hierarchy. And instead of saying, gate 1 and gate 2, what we can do is we can make a little hierarchy. We can say that if our gate 2 falls under gate 1, that's called an AND gate. And essentially that's what we do in a lot of our flow cytometry analysis, is we set up our gates, we put them into these little hierarchies, and that way we can say, all right, well, let's look at things that are lymphocytes. And then let's look at things that are lymphocytes that are CD3 and CD4 positive. And let's look at lymphocytes that are CD3, CD4, CD16, yada, 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 positive. So by using these gating hierarchies when we do flow analysis, it allows us to discriminate different populations and phenotypes of interest. Now, the one other kind of thing that goes into flow data, I think, that's pretty useful is being able to measure central tendency. Right? So when we look at a histogram, we kind of get this peak. And a lot of times we kind of have to define, where the median value of that peak is. And what that is referred to when you read literature and you read papers about this stuff, it's called MFI. It's called mean fluorescent intensity. Now, if you've already kind of gotten through some of your basic math classes and things like that, you might remember that there's different ways to measure a mean, right? You can measure an arithmetic mean, you can measure a median, you can measure a geometric mean, you can even measure something that's called the peak channel. And actually what happens a lot in publications is people refer to the measure of central tendency as MFI. And very often publishers never ask them, what did you use to measure that? Was it geometric mean, median, or arithmetic mean? So in flow cytometry, it can actually mean any of these different things. And when you are reporting your data, I think it's very useful to annotate what you're actually calling these things in the software. So I'm going to kind of move on here. There's this whole idea of this concept of something in flow cytometry called compensation. Honestly, I think I'm going to skip over it here just in the interest of time. But what happens is, if you're using one of these kind of smaller, handheld systems that just has 1 or 2 detectors in the system, what happens is that light just kind of goes into the correct channel and you make a measurement. But what also really happens there is some of that light from that FTSE signal can get into that other detector. So some of that green light can actually get past that filter and get into that red detector. And some of that red light can actually get into that green detector. And what we have to do with this tool called compensation with flow cytometry is essentially account for that, right? We need to know that the signals that we're getting from our cells of interest are, actually green CD4 or actually red CD8 without having any sort of overlaps. But the good news is, generally there's some very smart people running these flow cytometers, the people that teach you about how to use them, and the first thing that they're going to tell you is that you always need to come with a proper set of controls. What that usually means is a tube that's only been stained for one color. If you're running a 2-color experiment, a tube that's only been stained for one of the colors and another tube that's only been stained for another of those colors. And really, I think the long and short here is as long as you have those controls, you're going to be able to run your experiment effectively very well. So again, I'm going to kind of skip through some of the compensation stuff here because it's a little bit more technically involved, and I know with the system that you're going to be using tomorrow, it's going to be a 1 or 2 detector system, so you're not going to have to really worry about compensation. And that's another really great thing about using a smaller 1 or 2 detector system is because you don't have to worry about those kind of caveats. So what we're going to do now is just take 5 or 10 minutes to actually analyze a few data files and show you what that looks like from a scientist's perspective. Data comes off of an instrument, you get this file, well, what do I do with it, right? And when we work in the software tool that the company that I work at makes, the kind of big difference between us and other tools that are available out there is because we kind of look and feel and work like Microsoft PowerPoint, and we wrap in kind of these extraneous tools that people generally have to export to into one software package. So just kind of like, I don't know if this generation has had all of these kind of technologies in the past. Like, when I was in college, I actually had a GPS and a cell phone that probably looked exactly like this, and a camera, and I always carried them around in my backpack. back, right? Now everything's wrapped into my smartphone, so life is really easy. And the same thing can be said of flow cytometry data analysis, where you can do all the things that you can do out in Excel and PowerPoint directly in our software package that we make, so that way you don't have to carry around all these kind of extra tools and things like that. So the two kind of quick use cases I want to go through are live dead cell discrimination, and I think this is something that you're going to focus on tomorrow when you actually acquire some data, and then just a simple CD4 or CD8 staining. All right, and it really is pretty easy in FCS Express. So let me open up this data analysis tool that we call FCS Express. And I know it looks just like PowerPoint, right? I'm sure everybody here has probably worked with PowerPoint at some point. And in PowerPoint, you do things like insert a shape or text box or a picture, and you kind of build up a presentation for what it is you want to communicate to folks. And we can do the same thing, but with flow cytometry data files in FCS Express. So let me get some of this other stuff off the screen here, and give it a second here. Let me get this stuff out of the way, get our actual PowerPoint out of the way, all this other stuff. And remember what I mentioned, that the flow cytometers create an FCS file, right? This is what that looks like. A flow cytometer will create a series of FCS files for however many tubes that you've analyzed that day, and it will put them into a folder. Just like if you took a picture on your camera, it's going to put them into a folder with a whole bunch of different, file types for that. So if we want to get that data, say off of the instrument where we acquired and into a software package that's very easy to analyze it, well, what we can do is kind of like PowerPoint. We can drag and drop a data file, an FCS file, into FCS Express and ask it to insert some sort of representation of that data for me, like a density plot, or I'll put it in a histogram as well, right? And kind of like PowerPoint, you get objects on the page, and you can move them, and you can resize them, and kind of get them wherever it is that you want. And in this experiment, again, this is a live dead staining. And if I change to my fluorescent parameter, which is the amount of propydian iodide that's been either incorporated or not incorporated into the cell, I can visualize what's going on. And when I talked about gates, the way that we kind of handle gates and what we call markers on histograms is we can just draw a little marker. And here, if you remember correctly, things that get a propydian iodide that stains are going to be things that are dead cells, right? So I'm going to label those dead cells. And things that do not incorporate propydian iodide, I'm just going to put over here, I'm going to call these live cells. So for this one particular tube, I know I have 94% of my cells are alive and 4.5% are dead, right? But I don't just have one tube in this experiment, right? In a real world scenario, I'm going to collect lots and lots of tubes. I'm going to collect replicates. I'm going to do everything really properly. I'm going to look at, I treat the cells, I don't treat the cells. And what FCS Express allows you to do is use this little button here to kind of quickly navigate through your data files, right? So I can go through and I can say, all right, well, this tube has 10% dead and 87% live, right? 88% live, 9% dead. And I can kind of scroll through and I can say, oh, look at this one, right? Nothing died. What do we do with that? That's pretty important, right? That's probably a good one to go back and look at. So by using, analysis tools like this, we can quickly crunch through lots and lots of data. And it doesn't mean that I need to sit here and write down that that tube had 98% of cells and then move to the next one and write down that that one had 99% of cells, right? There's all this kind of functionality in the software that I don't want to really bore you with today about how we can export this stuff to Excel and get it into spreadsheets in FCS Express. So instead of having to go through and write things down or copy and paste and move back and forth, you can actually just get a kind of full collated report of all this information by clicking a button or two in the software, which when you become a scientist, that's going to add kind of time back into your day, and time is important so you can run more experiments, get back on the lab bench, and kind of do more things, right? The one other type of experiment I want to look at, and I'm going to wrap up with here in just the next 2 or 3 minutes, is looking at a CD4, CD8 staining, right? So again, I've got a few different data files here I've acquired on a cytometer. I'm going to drag that data file into here. And in this kind of data file, what I'm going to do is set up a few gates. You can see I've got a few different parameters here, CD3, CD4, CD8, CD19. I've got more than I actually want to look at. But I'm going to look at those side scatter and forward scatter parameters. I'm going to create one of those two-dimensional gates around this population here that I know is the lymphocytes, and I'm going to click OK. And I'm going to insert something that we call a gate view. And essentially, this is going to be that little gate, this gate hierarchy that I told you about. So if I want to say, of those lymphocytes, let's look at, some other subset of cells, like let's look at CD8 versus CD4, right? Anything that falls within this lymphocytes gate is what's going to be shown on this chart over here. And if I move this gate and I look at the monocytes or I look at the granulocytes, we get that information updated in real time in the software FCS Express, which is pretty cool, right? So we can actually see that there's some CD4 and CD8 in here. So if I wanted to kind of evaluate, all right, well, how much CD4 and CD8 is there? What do these population densities look like, right? I create another little gate here, and I'll call this CD8. And I'll create another little gate here, and I'll call this CD4. And let me drag this over here, we'll give it a different gate color. And you can see that little gating hierarchy is filling out, right? So anything that's lymphs, I'm evaluating here that are lymphs and CD8, or lymphs and CD4. And if you've got 2 or 3 markers, that's fine. If you've got 30 markers, you just keep dragging these gates out and changing to a different parameter and creating new gates and kind of doing that over and over and over again, until you drill down into the population of the phenotype of interest that you're interested in. And then again, like I mentioned before, just quickly navigate through the data, and you can kind of look at the changes and the statistical differences between these different treatments, between these different tubes, between these different cell types, and really kind of gather the information you need. Okay? So I think that's enough on an overview of flow cytometry and an overview of the data analysis today. What I do want to kind of leave you with is that there's a lot of resources out there, right? What I talked about today, it's literally the tip of the iceberg. Flow cytometry is a technique that's been around for many decades, and it's been really refined in just the last 10, 20 years or so to become even more powerful than it ever was. So look at the resources that are out there. We have one on our website called Flow Cytometry Basic Introduction. Essentially, this is a free book. You can go to the website, you can download it, but it also provides real-world digital data for use within FCS Express, where you can go in and you can get access to data files like I showed you. The other thing I'd recommend is, as you're kind of, working your way through labs and courses and doing internships and things like that, if you see somebody that has a flow cytometer, ask them about it, right? It's honestly one of these tools. When I came out of college, I knew a little bit about it, but I saw it everywhere I went. And I think it's a really great thing, if you can even just have it on your resume, that you understand a little bit about what a flow cytometer is, that you know what it does, you know the importance in the field of science. Hopefully that will help you down your kind of career path and your journey to kind of become a scientist in the future. So with that, just want to say thank you for your attention for the last hour. I wanted to see if there's any questions that folks have. I'd be happy to address them all on the line here. If not, you can contact the course instructor. You can contact myself or our team at support at De Novo Software if you ever need any help with data. So any particular questions, comments, things I can address?
Thanks, Sean. I'm sure there's a lot of information for the students to absorb, but maybe let's see, does anybody have any questions? Any general questions about any of that? Nothing yet? They may need some time to absorb some of this information before they can ask.
Absolutely, and I think when you're actually acquiring samples on that cytometer tomorrow, it's useful to see the theory, but it's also a lot more useful to see the theory in practice, right? When you actually have one of those handheld cytometers, it becomes very visceral.
Yeah, I think we'll have some fun with that tomorrow.
Excellent.
Yeah. Well, thank you for your time. I really appreciate you spending the time with us. The students are giving you a little round of applause in here. I don't know if you can hear that. So thank you, and hopefully we'll be in touch soon. And George, I'll see you tomorrow.
Yeah, I'm looking forward to it. And certainly if your classes have some questions, I can help to answer them. And we'll run through a little bit of this as well tomorrow prior to the Yeah, and and we're on for tomorrow at 10, correct?
10 o'clock we start.
Great, I will be there.
Okay. Thank you.
Thank
Bye,
you.
everybody.
Take care. Bye bye.
Thanks. Bye.